These methods can help answer key questions in the field of biomaterials, particularly for bone regeneration. They are used to evaluate the effectiveness of biomaterials and potential immune responses. The main advantage of this multidisciplinary platform is that it provides a range of parameters for predicting biocompatibility of biomaterials used for bone repair.
The implications of these in vitro and in vivo bone techniques extend towards other bone disorders due to the need for preclinical screening of novel orthopedic biomaterials.
Though the immunological methodology can provide insights into biomaterials for bone healing, it can also be applied to biomaterial assessments for any other clinical indication.
Aspirate culture medium from primary osteoblasts 14 days after the addition of osteogenic mineralization medium. Rinse twice with 0.5 milliliters of 1x PBS. Then fix the cells by adding 0.5 milliliters of 10% buffered formalin solution and incubating at room temperature for 10 minutes. After 10 minutes, aspirate the 10% buffered formalin with a single-use pipette and wash twice with 0.5 milliliters of ultra-pure water. Then add 250 microliters of 40 millimolar alizarin red S staining solution to the fixed osteoblasts and incubate the plate at room temperature for 10 minutes on a shaker at 100 shakes per minute. Following the incubation with staining solution, aspirate the staining solution with a single-use pipette and rinse with 1 milliliter of ultra